Oh, great. Great. So I have the pleasure to introduce Wengong today, who is a postdoctoral associate at the Wendy Smith Center at the Broad Institute. So he finished his PhD at MIT CSL by Regina Bozzi and Tommy Giacola. And his research seeks to develop novel machine learning algorithms for biology, including drug discovery, immunology, genetic engineering, and synthetic biology. And so today, Wengong will be giving a talk upon his iterative Refinement graph neural network for antibody document design. Before we start, a few housekeeping businesses. Now, we've had an unfortunate spate of Zoom bombing in the past 2 weeks. So for this seminar, we will be keeping chat and video off for the entire duration. Now, in the last 10 minutes, we will make chat available again. So if you can hold any questions that you have until the last 10 minutes and then put them in the chat for when going to address, that would be appreciated then. But without any further ado, Wengong, you have the floor.
Oh, thank you. Yeah, thank you for inviting me here. Today I have I will talk about my recent work on using deep learning, deep neural networks for antibody modeling, especially for immunology applications. So antibody design, the goal is to design high affinity antigen binders or neutralizers. Antibodies bind to like viruses or these viral proteins in order to trigger immune response or block this infection pathway. And its therapeutic utility is huge. It can be used as antivirals and many other utilities. But its therapeutic efficacy is usually defined by the binding affinity to the antigen. For example, some of the antibodies may bind to the antigen very well, but some of them don't, right? So you want to design some antibodies that has good binding affinities. Just to clarify a bit of terminology here, antibody community kind of have these several words. One is the CDR, this complementarity-determining region. It's sort of at the tip of these antibodies, these Y-shaped proteins that has, that is primarily responsible for binding to the antigen. The part that binds to the antibody is called, on the antigen side, is called epitope, which is called epitope, right? So today I will kind of use this cartoon visualization for antibody. Basically, the top side is a CDR structure, and each circle is a residue. Each residue is basically, has different type of amino acids, and the bottom parts that with these arrows indicates the framework region of this antibody variable domain. I will not go into too much detail about the background of what antibody how it's composed of, but these are the terminologies that we will primarily use today. So basically, the antibody community focuses a lot on the design of the CDR, again, the tip of the antibodies, which is most variable part of the antibody, and it plays the central role of binding. So, okay, cool. So this is sort of the short background. I want to kind of focus more on the machine learning side of today's talk rather than going through many biochemistry backgrounds. So I want to kind of, in the beginning, want to highlight some potential applications of deep learning in immunology and also especially antibody engineering. And there are several questions that could be automated by machine learning. One is the prediction, obviously. Given an antibody, can we predict its binding affinity to an antigen? This is, you can sort of represent the antibody either as a sequence or a 3D structure, and this is the prediction side of the picture. They also include stocking, which we will talk about today. Is if I have a antibody and a antigen, can I predict how they interact in terms of how they interact in 3D, right? Basically, what are the shape of this antibody and how it's located inside of this antigen epitope. So that's also part of the prediction. There's also the design part of the work. The design part, right, which is the inverse of prediction. Basically, you want to design an antibody sequence, especially the CDR sequence, so that this antibody will have a high binding affinity. Sort of the example workflow that I could imagine, there are many different ways of formulating this antibody design workflow. You can think of this as the following loop, right? For example, let's say we have some antibodies that already binds to some antigen but the binding affinity is weak. Then we can sort of optimize the CDR of this antibody so that it can bind better, right? So we can imagine this workflow with two components. For example, we can use a generative model to diversify the CDRs of one antibody, maybe coming out of some experimental screening, right? Then we can diversify the CDR, construct a library of potential antibody candidates, right? Then we can use another model, which is docking model, to kind of dock these antibodies, especially the CDRs, on top of these the selected epitope, and then kind of based on the docked structure, kind of see what's the binding affinity and select the optimal sequence, etc. Right? Of course, this is not the only way to formulate antibody design, but it's a is a useful formulation that we are exploring, and we are collaborating with a variety of biologists at Broad Institute and Koch Institute for various applications. I guess so. Today's talk, the key is to kind of address how to use machine learning to kind of to build these generative models or these docking models. So this will be so today's talk will have two parts. One is this design and the other is docking, all right? So let's go through the first part, basically generating these CDRs that is compatible with a given antibody framework. All right, so let's see, okay. So just to mention some prior work, right? Generating these, so there are quite a few work already that does kind of the same thing, generate antibody CDRs using generative models. They are primarily based on sequence-based. They are primarily sequence-based. So to be specific, we can prior work used recurrent neural networks to generate these CDR sequences, and right. And so it's the same as how you generate the text, right? You just generate residue by residue. I guess this is straightforward. So if you don't know what recurrent neural network is, just in case, then we can definitely talk during the Q&A session. But at high level, right? Let's say you don't need to understand what the mechanism is inside of a black box neural network. But you can already see, right? This model does not, the caveat of this model is it does not take into account the shape of the generated sequence. So in a way, the shape is important for modeling antibody-antigen interaction. As you can see from, this is the figure that we showed in the earlier slide. You can see that some antibodies are good because the shape matches the antigen, while the other don't, right? So in some sense, incorporating the 3D structure in this generated model is something is quite important, and it will definitely help the performance. And in fact, this is not new, right? There are like these inverse folding models that is conditioned on the backbone structure. And previous work has already shown that if you know the backbone of the protein, let's say you have this chain here, you have if it's backbone shape, if you just given condition on that backbone 3D structure and generate these residues one by one, then you will get a much better log likelihood. Here, perplexity is just another way of saying log likelihood, and lower perplexity means higher log likelihood. And you can already see the improvement with incorporating the structure is huge, right? But again, this framework is not ideal because you assumes the 3D structure is given already, but in many of I would say in a variety of applications, you don't know the 3D structure before you start, right? So it's like a chicken and egg problem. You have the structure which helps you to find a better sequence, but you actually need to know the sequence in order to know that structure. So I guess so. The question is, how can we kind of go around this chicken and egg problem? And this is what this is our proposal, which is called sequence structure co-design. Basically, we want to design a generative model over both the sequence and structure, right? Instead of assuming that you know the structure beforehand, but we want to actually generate the structure and the sequence at the same time. So it is kind of viewing the 3D structure as a latent variable while you're designing a sequence. And while you're designing a sequence, you want to kind of leverage the structure information, this latent structure information, as much as possible in order to find a better sequence. So ideally, we want to find for example, if the sequence-based model is like this, if you know the structure-based approach performance is here, then ideally, if we model the structure as latent variable, we want to find a sweet spot in between, right? So that's sort of the hypothesis, and I will show the results later to verify whether this is true or not. I guess based on this formulation, right, this co-design formulation, then how do we actually implement this idea? And now, since we have two variables, right, the structure X and the sequence S, and they are intertwined together, right? So we need a single data structure to describe these two variables together. And this is what we do, right? So we represent a sequence and its 3D structure as a geometric graph or a point cloud, right? Based on the residue coordinates. So this is a k-nearest neighbor graph representation of the CDR. I'm skipping the the framework region for now, but this is the basically you have a bunch of edges, and each edge has some features like the distance between the two residues, or what's the direction or orientation between the two residues. I will not go into the details, but this is the rough idea, right? So basically, you are generating a geometric graph to realize the 3D structure and the sequence at the same time to execute this co-design idea. So and how do we generate this geometric graph? Again, there are a variety of ways of generating these geometric objects. I think I think all of these sort of geometric generated models are trying to capture sort of one idea is you can actually iteratively morph this point cloud into the right shape. And this is one of the approach that we took that I will utilize today. Is called iterative refinement. Basically, well, so here's the thing, right? So this shape, this point cloud itself is continuous, but the residue, the amino acid itself are themselves are discrete, right? So how to kind of reconcile the two is important. And here our formulation is, our solution is, we generate an amino acid sequence kind of still one by one, right? This is the same as these recurrent neural networks, but we do need to utilize the structure, right? So how do we do this, right? So every time when we insert a new residue, then we need to update its structure, and it is not just updating this residue, the new residue being inserted, but we want to update the structure globally. And this is important because imagine from the physics point of view, you insert one residue and it's not gonna be like everything else is gonna be static, right? They will all change correspondingly to accommodate this new residue. So that's why we, once we add our residue, we will update its structure, kind of, update the coordinate of each point, each residue, and we keep doing this iteratively. So this global update will allow us to, capture this, to better incorporate the 3D structure while generating a sequence. So just to go into a bit of detail of how to execute the idea, right? So first we will encode the CDR structure by a graph neural network. Again, to simplify this, let's skip the framework region for now, right? Let's just look at the CDR, right? So you have this graph, right? So you can encode this using a standard graph neural network. Once you get this vector for each residue, I should have said we will first guess what's the number of residues in the beginning in order to so that we can update this structure kind of globally, right? So you will initially everything will be blank and you are filling in the residues one by one. So suppose now we are at the 5th residue, right? We apply this. So each empty residue, it has a special is a special token, basically a mask, right? A special token, and you encode this graph using this graph neural network, and you get this vector. Then you predict what's the next residue, right? So this is basically a classification task. You have 20 different amino acids, and you just do the softmax. So then you can select the select the residue based on the prediction, and then we need to update the geometric graph kind of, to do this global update, right? So what we do is we predict the C alpha coordinate of each residue, and you basically get this xi, right, for residue I, and you predict this again from the same this representation learned from this graph neural network. But I just want to emphasize that our loss function is rotation translation invariant, even though we kind of output this coordinate directly. And because our loss function is actually over the distance rather than the coordinate. So we are not predicting the coordinate directly, but the model kind of just picks one coordinate system by itself, and then and everything within that coordinate system, the distance will so basically the loss function will train the model to produce these coordinates so that they will be close to the ground truth coordinates from the crystal structures. So the learned coordinates will be equivalent to the ground truth up to rotation and translation. And yeah, so and then this is one step of update, and you do this, iteratively, right? So you until the model until you fill out all the residues. All right, so this is the main idea of this this generative model. But there are some additional like components that we want to incorporate, right? So for example, we have this this framework region, which actually provides some level of information of what kind of CDR residues that you can put in here, right? And so how do we do this framework conditioning, right? So what we do here is we can, still operate on this iterative refinement idea, but extend this graph to a joint graph over the CDR and the framework region, right? So basically, we have each edge describing the distance or the orientation between the two residues. But it could be both. The residues will be will incorporate both the CDR residues and the framework residues. And this joint graph representation, what it's trying to capture, right? It is trying to capture how the framework region is kind of interacting with the CDR residues. And we want to make sure, right, when you insert these CDR residues, it should be kind of compatible with the framework region, which is given, right? So again, the idea is the same. It is just we have more conditioning, right? We condition on not only the previously generated CDR and the current CDR structure, but we also condition on the structure of the framework. And we do this iteratively, right? While we are filling in the missing residues. So this is sort of I would say, yeah, almost identical to the existing the CDR only version of the model, except that we need to do a little bit of acceleration. So basically, we well, for CDRs, it's usually just around 10 to or 20 something residues, and this graph has just 20 nodes. You do this iterative refinement. It's not too expensive. But once you start to do this iterative refinement over the whole variable domain, it's a little bit slow, right? Because you now have more than 100 residues, and they are all needs to kind of move while you're updating the CDR residues, right? So I think that this is an interesting question of how can you do sort of coarse graining over the protein structure, so that it does not lose much information while accelerating the folding or design. Here we took a very simple idea, basically group bunch of well, because the framework region, the sequence is given, and they are more or less constant, right? That's why we are not really designing a CD. We are only designing a CDR, but not the framework, because the framework region of an antibody is kind of constant. You pick a bunch of antibodies, their changes are relatively small. So based on this sort of intuition, what we can do is we can kind of group the residues in the framework, kind of so that you, for example, you can construct these k-mers, right? For example, if k equals 4, then I can just basically cluster each four basically put 4 residues together into one block, and you do this for 4 by 4 or 4 by 4, right? So this sort of picture here illustrates this idea, and this will reduce the number of residues by 4, right? So if you have 100 residues, now you have just 25 residues plus the CDRs, right? We don't apply this coarsening over the CDR because we're generating them one by one. So then we end up just having 30 something residues that you can do this iterative refinement quite quickly. Just to clarify, right, what do I mean by the coarse? So what do we do with these coarsened blocks, right? The embedding of this coarsened block will be the average embedding of each amino acid, right? For example, here you will have the embedding of the of these four, and you take the average for the coordinate of this block. It will also be the average, basically the center of these four residues. So then you apply this iterative refinement to fit the right shape and also fill out the right CDR sequence. Right. So I know that this kind of presentation focuses more just on the high level ideas, and I'm definitely happy to talk about more details offline after the whole talk. Let's look at some evaluations just to see what is working and what needs to be improved, right? So we trained this model on 4,000 crystal structures from the structural antibody database. And we split the training and test set based on the CDR sequence similarity. Some CDRs, they are almost identical to each other. So we need to do this sequence similarity split to ensure that your test set is kind of meaningful. It's kind of new, right? So we did this, training test split. We do a bunch of evaluation. First, we want to look at whether the predicted this shape matches the matches the CDR shape. Does it match the ground truth? And we calculate the root mean square error of between the ground truth and the predicted structure, and you see that our model kind of outperforms. So we made a baseline on basically not doing this iterative refinement, but rather just generating. This baseline. I will talk. I can. The details are actually described in a paper, but the basic idea is to follow these autoregressive graph generation, existing graph generation work that kind of just fill out the distances just one by one. So it does not do this global update, and which basically has a lot of basically you cannot correct the error that you made in the past. So this model kind of performs much worse than this model based on iterative refinement. And you can see here, if I do not use this iterative refinement idea, but rather kind of predict one distance and then just fix it and then move on, then you will see that the model will actually generate some weird shape if you don't kind of correct if you don't allow the previous residue coordinate to be updated after you generate new residue. So this is on the structure evaluation part. There's also the log likelihood part that we want to look at, right? Because we are actually, this is a generative model, and we want to know if the generated sequence actually, makes sense. And one way of, defining making sense is using this log likelihood. Basically, if my model would assign high probability to the real antibodies in the database, right? So here the evaluation metric is perplexity, right? So and you can see that if we compare with the sequence-based LSTM model, which is used by the prior work to generate the CDR sequences, you can see that our model outperforms the sequence-based approach because we utilize the structure on the fly. And as I mentioned earlier, right? If I know the ground truth 3D structure, it will obviously help. And you can see here the perplexity is much lower. And because the 3D structure is given, and the 3D structure will constrain what kind of residues that could be there, right? So you can see that our model kind of performs between the two, right? We have the sequence-based approach that ignores the 3D structure, completely, while this we basically kind of, we are kind of in the sweet spot between the two. That's because we leverage this 3D structure as a latent variable. So this is, oh yeah, maybe I will, I also have some additional ablation studies kind of showing, how if, how do, if, there are various ways of conditioning and do this framework conditioning, but I think for the sake of time, I will kind of skip this part. Now coming to another evaluation, here we want to actually compare with Rosetta antibody design. And we want to see if our model based on this data-driven approach, can we, how does it perform compare with existing, physics-based models? And you can see that here, so we kind of evaluated our model on 58 antibody-antigen complexes, which is, so basically this Rosetta antibody design provided they selected they selected a benchmark data set around like 60 antibodies, and they proposed this evaluation metric called sequence recovery, which is indeed a standard metric among the protein engineering community. And let's see if, so here, just to clarify, right? Sequence recovery means the percentage of the residues that matches the native sequence, the ground truth in the crystal structure. So you can see here on the right that our model, called RefineGNN, it does actually achieve a higher sequence recovery rate compared to Rosetta and also the other generative model-based approaches. So this is sort of the first part of the sort of this workflow, right? We can generate these CDRs, kind of capture the real distribution better. But again, this sort of generative modeling part does not really incorporate the antigen structure, which is actually a big caveat. You don't, to make this process actually biologically relevant, we do need to incorporate whether this, how does this CDR interact with the antigen. So ideally, right, I would say if we can have a generative model that can also condition on the antigen and kind of do this, also incorporating the structure, it'll be great. Here, let's focus on a sort of simpler task, right? If I have, let's decouple the generation from this structural modeling a bit. Let's say I have a CDR sequence, and can we actually predict how it interacts with the antigen? And this problem is known as docking. And let's see if we can use if deep learning models could actually perform these kind of docking tasks. And of course, there are already a variety of work that's trying to address this task, right? For example, this well-known work from DeepMind, AlphaFold Multimer, also tries to do this docking, but it assumes the epitope is unknown and none of the 3D structure is known. So you start from two sequence and let's say an antigen sequence and a antibody sequence, and then just fold it. And the DeepMind paper itself claimed that AlphaFold Multimer does not work for antibodies in general. And we tried on a couple of examples, and indeed, it does not fold into the right place, especially it does not locate the right epitopes. So based on their claim and our sort of investigation, we decided to kind of make this task a little bit simpler to see if, for example, let's assume the epitope 3D structure is given. We note which part of the epitope, which part of the antigen that we want to dock into, but we still keep the CDR or the antibody as a sequence without knowing its structure. And so basically our formulation is the following, right? We have a CDR sequence and a given antigen epitope in its 3D structure. can we predict its shape? Sorry, its docked pose, right? And if we can predict this docking pose accurately, then we can use it to score different CDRs based on their binding energy and etc. So this is sort of the goal that the task that we want to solve. So let's look at how to extend the previous model, this RefineGNN model, to kind of address this task, right? Here, there are two additional challenges that we need to solve. We can, unfortunately, we cannot just apply this iterative refinement directly. Well, we will operate on iterative refinement, but we need to add some additional components in order to make this complete. So first, we need to fill out the side chains. Again, right? The previous model, we just look at the backbone of the CDRs, and we're not really modeling its side chains at all, right? But side chain contacts are important for modeling antibody-antigen interaction. And also we need this equivariant modeling. This RefineGNN model that I showed showed before in the first part of the talk, it predicts the 3D coordinate directly, right, using this linear transformation from the hidden representation learned by the graph neural network, right? So it basically learns its own coordinate frame. Everything is in this coordinate frame. Their relative position are right, but it does not. So everything is right within the its own coordinate frame, but it does not, change. It's not compatible with the antigen coordinate frame, right? The epitope or the antigen can rotate arbitrarily, and the RefineGNN model will still just predict the same coordinates in its own coordinate frame. So this is a problem, right? So ideally, we want to predict the coordinates of the residues in a way that is equivariant to the antigen. So that's the idea, and these are the two problems that we need to address in order to kind of extend this RefineGNN model for antigen docking. So just as a side note, why do we stick with the coordinates? How about just predicting the pairwise distances, which itself is so basically you can imagine that I can just predict the pairwise distance between the two residues, either within the antibody or across antibody and antigen. So the reason why we want to predict the coordinate is because pairwise distance actually loses quite a bit of information, especially chirality. And this has been addressed in a variety of work on previously. So basically, by chirality, what I mean is, for example, if you just know, let's say we have 3 points and you have 2 distances, right? So you just knowing these 2 distances, it does not tell you whether these 2 residues are on the same side or on the opposite side of the residue Si, and that actually is crucial for many these protein design or folding or docking applications. So that's why we want to operate on the coordinates, which captures all the information that we need, but we do need to address this equivariance challenge. So let's go through this model kind of step by step. So in the beginning, we will initialize the CDR. We need to, so the input to the model is the sequence, right? The antigen is in its 3D structure. All the coordinates are known and fixed, but the CDR sequence, you actually need to realize the coordinate in the right place. So starting from this sequence, we need to first initialize the coordinates, and we do this just by predicting the pairwise distances. And from the predicted pairwise distance, we can actually realize the coordinate of each residue. And this pairwise distance prediction is implemented through by getting the representation for each residue, right? For the antigen side, we run this graph convolution on the antigen to get these embeddings for each antigen residue. For the sequence CDR, we just run a sequence like a recurrent neural network to get this initial embedding. And we basically predict the pairwise distance based on these two vectors, and you get this pairwise distance, and you get from there you can actually get the coordinates. So now after you have initialized the coordinates, right, you would and you actually need to further refine this. Well, the initial prediction will have a lot of errors, and that's why you actually need to kind of morph it, kind of refine it so that it will go into the right place. So now we haven't really addressed the side chains really. So now what we need to do is to do this iterative refinement over the CDR residues. So here, what we do is we need to get the encoding for the CDR residues, right? Based on the current. So let's say now we are at the step T, and we have all the CDRs and their current, based on their current coordinates, will again, construct this graph across the antigen and antibody, and we can run this graph convolutional network to get the embeddings of each residue. And we also need to predict. We also need to get the encoding for the side chain atoms, not just the backbones, right? So here, what we do is we will have two graph convolutional networks. One is operating at the atom level, just everything is just atom. And for example, let's just look at the subgraph here. We have a bunch of atoms. We have their graph structure, and based on their distances, we can run graph convolution to get these mini vectors for each atom. We have the residue level graph convolution, which is the same as before. You basically have each residue and their neighbors, and you just aggregate their representations. And we actually, we do a little bit extra. We actually propagate the side chain atom vectors, these representations, to this sort of residue level graph, because basically if so these side chain atoms, these vectors will get averaged to provide a low-level resolution encoding for this residue F here, right? So basically you will have two-level graphs. The residue level graph, you do graph convolution, but you also have a lower level atom level graph that also running graph convolution, and you get these atom embeddings and the residue embeddings. And from there, what we need to do is to move them, right? So we need to apply to update the structure, right? And you need to update the coordinate of the backbones and the side chains in this step for all residues in the CDR, right? So and again, right, we cannot do this linear transformation, which will not be equivariant. So what we do is actually something like this. So we instead of directly predicting the coordinate, we instead predict a force between the two residues. For example, here we have this phi x, and which takes two vectors as input. One is hi and hj, and they are the hidden basically the representations learned representation for each residue learned by this graph convolution, right? So, and we this is a scalar, and we multiply it with this direction vector xi minus xj. So it basically gives you this force, and basically this residue will push this residue further apart, or actually draw it closer, depending on the sign of this force, right? It could be either positive or negative. And the good thing about this force is that it is equivariant. If you rotate the epitope, right, this xi will rotate accordingly, right? Because now this phi x is just a scalar. So this bypasses, this addresses the issue of the, and this lack of equivariance. And we basically update this. So we apply this force, we predict these force and update the coordinate of all residues. And that's how we apply this iterative refinement. So so now we want to do some evaluation, right? So we want to first see if I have this docked structure and compare its ground truth structure, how would that look like? So here we again use the this benchmark data set from the Rosetta antibody design, and we take the top 50 antigen residues as our epitope. So basically, given a CDR3, we just look at all the residues that are on the antigen that are closest to the CDR3, and then we take the top 50, and that gives you the epitope. And we want to find basically dock the CDR sequence to the right onto that epitope in a correct way. So here we basically after we dock a CDR sequence, we want to see if this thing agrees with the ground truth or not. And we use the dock Q score as an evaluation metric to see if the two agrees, right? So the dock Q score is between 0 and 1, and there are 3 stratifications. If it's above 0.25, it is acceptable quality. If it's 0.5, then it's medium quality. And you can see that our model actually could produce around 95% acceptable quality docked closes, but the medium quality is still a little bit low, around 17%. We also compare to see whether this model is actually working. We apply this. We also compare with another method called HDock, which performs this protein-protein docking, and we can see that our model actually outperforms HDock quite significantly in terms of the dock Q score and the number of acceptable docked poses, right? So on the left-hand side, we have an example of docked. This red one is the docked CDR, right? And the green one, perhaps it's a little bit hard to see. It's the ground truth, and we the two kind of roughly agrees. Right. So another sort of ablation study that we want to see is what if we do not do this iterative refinement? How much does this iterative refinement gives you, right? And here we kind of we want to take a look at whether we recover the right contact or not, right? So if the two residues two residues form a contact if their distance is within defined threshold. Here we use 4 angstrom, and basically you have some pairs that forming a contact which is positive, and some don't, which is negative. And we see if we apply this iterative refinements or not just doing this initial distance prediction, what's the contact accuracy. And you can see that this iterative refinement actually improves the contact prediction accuracy a lot in terms of AUC. So this is sort of the kind of a summary of today's talk. So just forgot to mention, this docking part is still kind of ongoing, and we're hoping that, by integrating the two models, that we can, kind of do this virtual, this AI-based antibody CDR design in an automated fashion. And we are applying this to several biological projects. But again, there are many. There are a lot of things that are not perfect. For example, how to generate the CDRs condition on the epitope directly, right? So here, for now, we are this generative model. It just proposes CDRs, while the docking model take care of the antigen-antibody interaction. But maybe we can directly condition on the epitope and figure out the right CDR sequences. We don't know, right? Another question is how to calculate the binding affinity, right? So far, we only docked, the CDR onto the epitope, but we don't know how. We still need extra work to actually to rank different CDRs and select CDRs that will bind well, right? And the other one is so far we are we're assuming that the framework sequence is given, but perhaps we could start from scratch. Well, we decided that we just design the CDRs because most of the variability or the binding affinity that does come from this part rather than the framework. But perhaps we want, as a cleaner version of the method, we may want to start everything from scratch. But there are a lot of basically there are still a lot of improvements that needs to be made, and this work is supported by a lot of collaborators from Broad, from Reagan or Koch Institute, and from MIT, from Sanofi, and from many other sponsors. Just want to say thanks to them for the support for our work. And with that, I can take a few questions. I'm definitely happy to stay longer if needed. Thank you very much.
Great. Thank you so much, Wengong, for a phenomenal talk. You can see the audience, but I'm just going to channel them and give you a round of applause here. I think we have a question in chat right now. So this is from Badon Bogdakis. I hope I pronounced that right. They say, great work. The sequence space is discrete. So how are you able to calculate derivatives in this discrete space? Can recurrent neural networks do that?
All right, that's a great question. So exactly because the sequence space is discrete, so we cannot we have to do this teacher forcing step, like the recurrent neural network. Once we predict a new residue, right? When we are predicting a new residue during training, what we do is we make a prediction, we calculate the loss, like but when we move on to the next residue, we just take the ground truth as a teacher forcing. So in that sense, we don't need to backpropagate through this discrete choice. Although I think an ideal way is maybe we can also do this iterative refinement over the sequence space as well. I don't know. I think that's an interesting idea. I think there are quite a few papers are trying to explore that in the context of natural language processing. But I think this is definitely a good venue to explore.
Great. I have a question about the coarsening model that you talked about, like where you took multiple amino acids and then summarized them into a single amino acid. So that looks like a straightforward averaging of the embeddings. And so I have some questions about that. I think that from from my perspective, it seems that some amino acids are going to be more important to the structure than others. Like I can imagine hydrophobic and charged amino acids being very important, whereas like stuff like iron or glycine probably isn't going to be very important. So it feels like this averaging operation really would mean that it's going to have to cut through that averaging rules and really focus upon like the important amino acids, the amino acids that are important on average to structure them. And so I'm kind of curious about how you think this is going to affect the embeddings of the amino acids, and it feels like it's going to have a pretty major impact that it's going to have to cut through this noise to represent like that chemo, if that makes sense.
Yeah, this is a great question. I think in terms, so right now, so again, right, this sort of coarsening just by k-mers is definitely a very crude idea. I think there are a couple of ways of kind of improving this approach. For example, we could align the frameworks with a bunch of existing antibodies and then see which part are conserved, which part is highly variable. Then we can, based on that, kind of merge only the conserved ones, but not merging the, the variable ones, right? This is one idea. In terms of more like these physiochemical properties like hydrophobicity or that, yeah, that's a great point. Well, I only have ideas about like based on the conserved or not conserved, but you can see the idea, right? You can do this dynamic clustering based on, well, maybe you can also cluster, say, if the two, if there is a big change, well, maybe we can cluster the hydrophobic residues together, but not hydrophobic hydrolyphic together. Some ideas like that would help, I guess.
That makes a lot of sense. Although if anyone else has more questions, please feel free to tweet them in the chat. I don't see any photo from others, so I'm just going to ask another question of my own then. I guess on a more higher level, I'm kind of curious about like, so in the second model, you showed that you need to know the structure of, I'm sorry, I forgot what it was called, but like what the antigen binds to, and then the structure of the antigen is unknown then. So I guess my question is that how common is the assumption that will on a particular like protein or that you want to target that particular like binding site for the antigen then?
Yeah, that's a great question. So it really depends on depends on how so again, well, these sort of projects there's always a biological project that's going on, and in that particular biological project, we know that it already binds to some part of the protein, but it's not binding that well. So you want to diversify the CDR so that it gets better. So in that case, you do know the CDR on the epitope already, and that's why the whole framework kind of makes sense. As you can see, whatever that I'm developing here kind of is operating on this optimization regime. I try to get something better. But if you say like starting everything from scratch, it is unclear, right, whether you would know the epitope. And I think we need to build an additional component of predicting what epitope would match this one, that matches the antibody. This is, well, the search space becomes much bigger, and I think this is definitely the next steps that I will address, but kind of building out components one by one slowly, and so that it will be more and more general purpose. So I think that's definitely a great, that's definitely a limitation of the current existing approach. And also a little bit biased because I came from the small molecule design world, which in most cases, they assume there is a binding pocket is given. But I do agree that in the case of protein design, it's actually a different story. So, yeah.
And suppose that you have the case where you may not know like a single binding site for that epitope, but you may like for example, have a few candidate different sites, would it be possible to iterate through a model and then test like each of those different like affinities for each of those different sites? And then is it at all possible to figure out like which of those candidate sites might be the best to target within antibody them?
Yeah, definitely. So enumerate, say, different patches and then see which one will be the best fit. That's definitely the straightforward way to go. Yeah.
Okay, that's the end of my questions, and I see that there's no further questions in chat. So I'm going to assume that we'll wind in to an end now. Thank you again for your great talk, Wengong, and thank you again for your time. And just another quick round of applause from me on behalf of the audience. Yeah, it was really a pleasure having you.
Well, thank you for inviting